can probably start.
Sure. Can you see my screen?
Yes.
Okay, great. Hello, everyone. I'm here today to talk about a part of the work that I did in my PhD, but I will not go into the details. I just wanted to mention a highlight that it was interesting for me. I hope it's interesting for you as well. So I'm talking today about polyphenols and how their diverse structures can be explained or can be distinguished using the LCMS. So first, I just want to introduce what are polyphenols. So polyphenols are a group of secondary metabolites. Actually, they are synthesized in plants. So humans and animals cannot synthesize them. And we can get them through our diet, mainly. And as you see, we have lots of benzene rings, we have lots of AH groups, and we have lots of substitutions. So that's why they are diverse, because if any of these groups are just changing their position, so it's a new compound or a different compound or an isomer of a compound. According to this, there are many subclasses of polyphenols. Mainly, they are flavonoids and non-flavonoids. I'm talking today about mainly the flavonoids. There are different, they have the same nucleus, but they have different substituents on the benzene rings around and different number of hydroxyl groups. So why these compounds are important or why people are interested in them? Actually, because they have some good effects, I can say. Mainly because they have antioxidant activities. So for the plant itself, they can protect the plant against the pathogens and any environmental factors like stress, temperature, drought, that might affect the levels of these compounds, and they might be synthesized more just to protect the plant against that factor. But for humans, actually, they have, because they have antioxidant activity, they can help as a cancer adjuvant therapy. They can work as antibacterial agents, anti-inflammatory. They have lots of effects, beneficial effects for humans as well. So to start with the analysis, since they are interesting compounds, we need to know how to analyze them. So there are actually many tools to analyze them. Today, I'm talking about the LCMS. Why LCMS? It's sensitive and selective. So it has two parts, actually, the LC and the MS. The LC is the separation tool. Actually, these compounds are present in plants in mixtures. So when we do the extraction, we have a mixture of these compounds. So we cannot actually know which compounds are there or determine their concentrations while they are there together. So we need a tool to separate them. So the tool that I'm talking about today, it's the liquid chromatography. So it has different parts. I'll not go into details. I'm just putting this for illustration. But there is an important part, which is a column. It has actually a packing material inside, and this packing material is interacting with those compounds in the extractor, in the mixture, and allow them to come out of the column at different times. That's how we have separation of these compounds. And then each compound coming from the column will just go to a detector to read. And then there is a software just interprets the signals that detector reads into peaks. So we can have some data or some figures to interpret and to know the quantities or the identity of a certain compound that we're interested in. So we talked about the separation tool. So let's go to the detection. The compounds coming out of the column should go to a detector. So one of the detectors, or the common detectors for measuring polyphenols, is the mass spectrometer. So how mass spectrometer works in simple words, we can say that all the compounds entering the mass spec, they should be ionized and they should be in gaseous phase. These compounds, after being ionized, will just go inside the mass spec, and then the mass spec can read the mass to charge ratios, which means that for small molecules, they usually have one charge. So it's usually reflecting what is called a precursor ion, or it's the parent ion, which means that it's the compound itself just ionized, will have a proton or just lost a proton. And then this compound will go inside the mass spec, and we might be interested to fragment that compound. If we fragment it, it gives different parts of this compound, and the mass spec can just read the ionized parts. We can think about it as a puzzle. So we have a compound, and then it just splits into puzzle parts. And these puzzle parts we can put together to know which compound is there for identification, for example. So this is an example. We have a compound. It's actually a flavonol. It's called camphor, and it has a glucose, a sugar attached to it. And when it enters the mass spec, it gives this ion. It's called the precursor ion. So it's the whole molecule with just one charge. And then when we fragment it, it gives two, for example, I just picked these two product ions, which are so unique for this compound. So this is just a spectra to show. So this is the first one, 447, the ion like representing the compound itself. It's called the precursor ion. And when it's fragmented, it gives different fragments of different puzzle pieces, as I mentioned. For example, this 285 and 151, and actually they are two major parts from that compound. And if we show these two parts, we can confirm that there was a control there. So after talking about the liquid chromatography and mass spectrometry, let's talk about how to use this sophisticated tool for analysis. So there are different approaches. We can use this tool for targeted analysis to quantify the compounds. So if we have standards, we can just quantify them. So we have the standards injected and the samples injected, and we just compare the standards to the samples and know the retention times and do the peak integration. Or we can use the same tool for identification. So we have unknown compounds, and we can inject our sample on this tool. It will give us some charts or figures, and we need to work on that to put the puzzle pieces together to get or to understand or to know the main molecule, what's there. So as I mentioned, there are two approaches. The targeted one, or mainly for quantification. There are two different types of quantification. There is the absolute quantification. It's using a standard, the same standard of the compound that we're measuring. Or there is something called semi-quantification. If we use a compound with a similar structure, because some of the compounds have no standards available. So the semi-quantification is the best approach available. So for example, if we have a cancel 7 glucoside, the one that I mentioned before, and we want to measure it, and we don't have the cancel 7 glucoside, but we have the cancel 3 glucoside, for example. So we can use that as a standard, and it's called calculating the equivalent, but it's not absolute values. It's not absolute quantification, but it's just giving idea about the amounts there. For the untargeted approach, usually we don't have standards, or if we have standards, we have them in libraries. So usually we have what is called relative quantification, which means that we just have ratio of intensity, ratio of the intensity of a certain peak to something else to normalize it. Accordingly, there are different levels of identification if we want to do identification. Sometimes we don't have the authentic standards. Retention time is tricky. People have different separation methods, and you cannot just compare the retention times of different peaks. And also how the compound is fragmented and you put the puzzle pieces together is tricky to figure out. So if you have standard, if you know the retention time, if you have the fragmentation profile, you can say that you go for level 1 identification. What if you don't have a standard, but you have a retention time and you have a fragmentation profile, so you're almost sure, like about to be sure that this is the compound, but you don't have a standard to compare to, you can say it's a putative identification. What if we know the retention time, we saw the fragmentation profile, we compared it to some libraries, but we are not sure. We saw some similarities to some compounds, but actually we're not sure that this is the compound. So we can say that, okay, it's a flavonoid, it's a flavonol, it's a flavone. We can just put it under a class. Or we're not able to identify, we would call it level 4. So usually in identification, we use a mass spec type. It's called Q-exactive. So it's a high resolution mass spectrometry. Why it's important? Because as you see, these two compounds, quercetin and hespertin, actually they share the same molecular weight. So when they go inside the mass spec, they actually give the same precursor ion. But as you see here, in their molecular formula, they are different. So when you get the ions or the data from your mass spec, actually, according to the number you get, you can predict the molecular formula. So there is a difference here in decimals in the ion that we got from the Q-exactive or the high resolution mass spec, which reflects that these are two different compounds. These are not the same compound. But if we use the low resolution mass spec, and you see, they will just give the same ion. So you cannot be sure if it's a quercetin or a hespertin. The highlight that I want to, I just give kind of a brief introduction through the tool itself and how to be used, different approaches. So I just want to give a kind of example that I've noticed in my work, that these are 3 different subclasses of flavonoids, anthocyanins, flavonols, and flavones. And if you notice that they have almost the same molecular formula, other than the first one, it has 1 more hydrogen, but they have different structures, totally different structures. And how to know which one is there in our sample if we did the LCMS analysis. Let's see. First of all, anthocyanins, these compounds that they have a positive charge, they are usually not stable, and they give the same ion when they are ionized in the mass spec, like flavonols and flavones. So let's see here an example. This is delphinidin as an anthocyanin, quercetin as a flavonol, tricetin as a flavone. And as you see, they are sharing almost the same molecular formula, but they have different structures. So first here, I'm just comparing the quercetin hexoside to delphinidin hexoside. It's quercetin with a sugar and delphinidin attached to a sugar. And I just try to compare their mass spec profiles without getting into much details. The main thing that I noticed is that for anthocyanins, or the compounds with positive charge, they usually show a water adduct, which means that the precursor ion is there, plus, and there is another ion with plus 18. This is a very common pattern for these compounds. So if we saw this in a mass spec chart, we just say that, okay, this is an anthocyanin without going further. We just go further to identify which anthocyanin is there. But for example, for a flavonol as quercetin, there is no this water adduct. This water adduct is not there. And if we fragment those two compounds, as you see, they have almost the same puzzle pieces. So this is a main or a major characteristic to differentiate these two compounds from each other. What about a flavonol like tricetin? We have also the tricetin attached to a sugar and quercetin attached to a sugar, so they are giving the same ion, but when they are fragmenting, mostly they are giving similar puzzle pieces, I can say, but we notice that the tricetin itself is giving a very intense fragment of 149, and a smaller one at 151 M by Z values. But for the quercetin, it's just giving 151. It's a very small detail, but actually it helped. It was the only difference to distinguish between tricetin and quercetin. And this is just the same compounds, but just they are not glycosylated. They are not attached to a sugar. So they show the same ions here, and even in the fragmentation, they show the same ions, but just different intensities. This is what I wanted to show today, that it's tricky sometimes to identify those polyphenols, but when you go into literature, when you have a good library, when you have a good amount of standards to compare to, you can figure out which compound is there. Yeah, that's for today. Thank you. Yeah, waiting for questions.
Okay, Oksana, are you going to stop the